Hello, it's now my pleasure to introduce Dr. Igor Bilinsky, Chief Operating Officer of Iovance Biotherapeutics. The format for this session is roughly a 25-minute fireside chat, and if you have any questions in the session, feel free to type them into the question box and we'll try to ask them on your behalf. So, Igor, Iovance recently completed its BLA submission for lifileucel in melanoma, which was an important step for the company. So maybe to start off, could you give us a brief overview of Iovance's TIL programs and the company's strategic approach to manufacturing, and then we'll dive into the details a little bit more. Great to have you.
Thank you, Gula. Good afternoon, Gula. Good afternoon, everyone. Great to be here. So Iovance, actually, I'll start with forward-looking statements. We're a publicly listed company. I'll be making forward-looking statements as part of this discussion. I refer you to our filings with the SEC for a full disclosure of risks and uncertainties. So Iovance, we're a late-stage immuno-oncology company with the potential to introduce the first one-time cell therapy for solid tumors to the market, TIL therapy lifileucel. BLA submission was completed last month for treatment of patients with advanced melanoma who have progressed on or after anti-PD-1 therapy. We are actively preparing for commercial launch of lifileucel, as well as for integration of Proleukin, an approved IL-2 product used in the TIL therapy regimen, upon the close of our planned acquisition of this product. We are very excited about our robust immuno-oncology pipeline and TIL monotherapy and combination regimens, including 6 cohorts of distinct non-small cell lung cancer patient populations across various stages of disease, a pivotal cohort in cervical cancer, a frontline melanoma phase 3 study, and the first ongoing clinical study of our first genetically modified PD-1 inactivated TIL. TIL are specialized immune cells that mediate tumor cell death, but may be suppressed and rendered ineffective by tumors or tumor microenvironment and suppressive mechanisms. Our TIL therapy platform is potentially applicable to many solid tumor types by leveraging patient-specific cells to recognize and attack diverse cancer cells that are unique to each patient. Cell therapies such as CAR-Ts typically act on a single or small number of shared antigen targets that are common to certain tumors and have been successful for treatment of blood cancers. This is a challenge in solid tumors where the majority of immune targets are patient-specific and fewer than 1% of neoantigens are typically shared among patients. Our TIL therapies are polyclonal and personalized to target a variety of neoantigens that are unique to each patient's tumor. We believe the polyclonality of our TIL therapies and our ability to centralize and streamline manufacturing differentiate us from other cell therapies and position us for success. We have built the first scalable centralized manufacturing process that is designed to make TIL therapies accessible for many patients and become a new major class of cancer therapies. We have built a significant amount of intellectual property, more than 60 patents, around our TIL therapy and manufacturing processes that further solidifies our leadership in the field. To date, we have treated more than 600 patients with Iovance TIL therapy manufactured using our proprietary process with a consistent manufacturing success rate greater than 90%. The process for most of our clinical studies and in our BLA submission is a proprietary centralized GMP manufacturing process that we refer to as Gen 2, which we use to reinvigorate and grow polyclonal patient-specific TIL recovered from a patient's tumor tissue into billions. While the TILs are unique for each patient, our Gen 2 process to produce TIL therapy and administer the treatment regimen is the same for each patient across solid tumor types. First, a surgeon procures a tumor sample that is shipped to our centralized manufacturing facility, where TIL are manufactured over 22 days into the final cryopreserved TIL therapy using our Gen 2 process to isolate, expand, and reinvigorate T cells. Upon meeting the product specifications, the cryopreserved TIL therapy is sent back to the patient treatment center. The treatment journey for the patient starts with lymphodepletion as a conditioning regimen, followed by TIL infusion and a short course of high-dose IL-2 to promote T-cell activation. The infused TIL migrate to the tumor site, where they recognize and target a multitude of tumor-specific neoantigens that are unique for each patient and harness the ability of the body's own immune system to fight cancer. When used alone, TIL is designed to be a one-time treatment. In earlier stages of disease, we're also combining TIL with standard of care, for example, with pembrolizumab. It is still a one-time treatment in that setting, but is accompanied by the standard of care treatment regimen. And then finally, on capacity, our teams are now busy preparing our internal manufacturing facility, Iovance Cell Therapy Center, or ICTC, for a potential pre-license inspection, while anticipating and planning capacity to supply patient demand at launch. Our internal facility is expected to provide most of the commercial supply at launch, while we will have flexibility to use our contract manufacturing partner to supplement clinical and commercial supply. I expect we'll dive into more details on this during the rest of this discussion.
Great, thanks for that overview. So with BLA submission now completed, can you discuss what you've learned from the process and how you hope to apply this to subsequent programs?
On the CMC side, we can leverage most of what we've learned and apply it to other programs. As I mentioned, the Gen 2 manufacturing process itself is the same process across solid tumor types. So we expect similar CMC sections in future supplemental BLAs. In addition, our potency assay matrix for lifileucel, which includes an innovative and proprietary co-culture assay, provides a template for our other TIL programs and other indications that use the Gen 2 process. The bulk of our discussions with the FDA over the past few years have been focused on CMC, in particular, the potency assays. So the FDA is now well educated on the topic and will be familiar with our manufacturing process and assays for subsequent submissions.
Got it. And more generally, what are the important analytics for this type of product? It's obviously coming out as a heterogeneous product from the patient, and you mentioned the polyclonality. So you mentioned the potency assay component. What are the important analytics that we should be thinking about?
So lifileucel is a first-in-class polyclonal T-cell therapy. We have been trailblazing in the manufacturing as well as analytical area. And lifileucel release assays include characterization of identity, potency, purity of the product, which is typical for cell therapies. What is unique to Iovance TIL therapy is our proprietary potency assay matrix, which is particularly novel as it is designed to reflect the polyclonal mechanism of action of lifileucel.
Got it. So you mentioned that you're preparing for FDA site inspection. Can you elaborate a little bit on how you're preparing and what else you are doing with respect to manufacturing for commercial readiness?
So preparation for pre-license inspections of PLI is an important focus area for us at this time. The PLI occurs during the BLA review process. In Iovance manufacturing quality and regulatory functions, we have a number of team members with recent experience of successful PLIs for several approved cell therapies, and also those who were formerly at the FDA. We are also supplementing our internal expertise with external consultants, including those who were FDA inspectors earlier in their careers. We have an expanded access program, or EAP, open, which provides access to lifileucel for patients in need and provides further experience working with TIL at the authorized treatment centers while we await approval. The EAP also enables us to further build these relationships with potential commercial ATCs, authorized treatment centers, and field test some of the key supply chain logistics, chain of custody, chain of identity processes that are optimized for commercial environment.
Great. And so for TILs, the starting material that you isolate from the tumor can impact the quality of the ultimate cell therapy product. Can you talk a little bit about the kind of training that you're doing at the ATCs with respect to sample collection that can ensure or maximize consistency with the starting material compared to what you saw in clinical trials where you've had the 90% success rate and the profiles that you've seen?
Great question. So a customized training program on tumor tissue procurement is part of the onboarding process that we are putting in place and have put in place for authorized treatment centers. That includes training surgeons on identifying appropriate lesions, appropriate anatomical sites for resection, for example, avoiding skin lesions due to the potential for bacterial contamination. Additional resources include lesion prosection after removal and prior to transferring into a tumor shipment kit. Everything from resection to sealing the collection kit happens in the operating room, which minimizes multiple people handling the samples at the treatment center. There's a paper published by Dr. Mullinax and other TIL KOLs in 2022 on surgical considerations for tumor tissue procurement for TIL, which was co-authored by our Iovance subject matter experts. And that paper includes, for example, best practices that minimize patient inconvenience and minimize patient morbidity while optimizing tumor tissue quantity and quality for TIL generation. And that's available it's out there published in the public domain.
Great. And so on the commercial manufacturing front, what size team have you brought on to drive that, and how is the training going there?
So right now we are staffed appropriately for BLA and BLA readiness activities, and we are building a team that we believe will be sufficient and of sufficient size to supply our forecasted demand at the time of launch. Hiring and training is progressing well. We are fortunate to have ICTC in the Philadelphia area, where we can tap into the talent with manufacturing experience at other companies in the region, as well as next generation of cell therapy manufacturing talent through local schools and resources. For example, we've just brought on a group of manufacturing technicians who participated in a skills initiative with the Wistar Institute, who are now became Iovance employees after passing the training program. It's an excellent opportunity for us to build a pipeline of potential local talent. And we're planning to have additional classes as we grow, and we hope this can also serve as a model for the industry.
Great. So you had a slide in your deck on the ICTC capacity. Maybe you can elaborate on some of those points a little bit. So how many doses per year do you expect to be able to produce at the ICTC at launch? And again, I think that was up on the slide. And kind of what kind of flexibility is there to expand, and kind of what are the potential timelines associated with that?
So the ICTC facility, as currently built, can supply 2,000 patients per year or 2,000 doses, with flexibility then to build out the shell space, which is part of that facility, to supply over 5,000 patients per year or 5,000 doses per year. Right now, we're staffing up the team to appropriately address our forecasted demand at launch, and the exact numbers for which we haven't disclosed.
But so in terms of being able to incorporate, let's say, additional capacity with CDMO, with partners, if you need to ramp up, what does that entail? And are there kind of timelines that you envision deploying that on?
Right. So beyond the initial launch, we will have access to manufacturing CMO capacity that we have in place, and that provides us flexibility to address near-term demand variations and fluctuations, access additional capacity immediately. To expand to 5,000 patients per year, the core building, the core structure at ICTC is already built. So the clean room and the build out would be shorter, we expect, than the 18-month that it took us to build the initial ICTC facility from the ground up. Beyond that, longer term, we have an option to a lot that's adjacent to ICTC, so we can construct an additional building there, and that may be necessary to get to 10,000 patients and beyond capacity, along with some automation initiatives that we have in place to further automate and streamline the manufacturing process.
Great. And what about manufacturing for ex-US geographies?
So ex-US ICTC is currently manufacturing TIL for clinical trials in Europe. Initially, we can serve Europe from ICTC commercially as well. And this was a key consideration for locating our facility in the Philadelphia area on the East Coast with good logistics and good connections to the airports. As we grow and expand into new geographies, we can consider adding new facilities, initially probably in certain parts of Europe or other key geographies with the critical mass of treatment centers and patients. But for now, ICTC, we expect, can serve the initial launch in Europe as well.
Mm-hmm. So beyond the current late-stage TILs that are using the Gen 2 manufacturing that you described, Iovance is also incorporating other innovations such as genome editing and TIL selection to some of the earlier programs in the pipeline, as well as we've talked about a shorter Gen 3 process. So can you talk about these different strategies and how you incorporate those manufacturing logistics?
So generally for genome editing, we're currently using the TALEN technology that we licensed from Selectis to inactivate specific targets, which is PD-1 in the case of our lead program, IOV-4001. We're now investigating IOV-4001 in the first clinical trial in advanced melanoma and advanced lung cancer. This manufacturing process follows a similar timeline to Gen 2, which is Gen 2 is a 22-day process, including the edit step for gene editing. We also have a shorter Gen 3 process, which is a 16-day manufacturing process that also enables us to manufacture TIL from a smaller amount of resected tumor, such as from a core biopsy. For example, the Gen 3 process is used in a cohort of the lung LEON 202 study for patients with the second-line non-small cell lung cancer, who in that cohort undergo a core biopsy rather than a surgical resection to obtain the starting tumor material for manufacturing. So our facility at ICTC was intentionally designed to accommodate these various next generation processes, whether it's Gen 2 or Gen 3 or a PD-1 knockout TIL 4001, which all use fairly similar logistics.
And I guess more generally, in terms of usage in the facility, how do you plan to use the space in the facility to continue to advance trials while also meeting commercial demand?
So we expect ICTC to supply most of the commercial demand at launch out of the core suites, while the flex suites will be dedicated to clinical supply. And then our CMO partners can provide us with additional flexibility for both clinical and commercial manufacturing capacity. And of course, our capacity planning accounts for our projections for both commercial demand ramp up and patient enrollment of our clinical trials. And so that's all part of the planning for the demand and the capacity that we're building to meet that demand.
Right. So in the current financial climate, there is considerable focus on costs for cell and gene therapy. So can you talk a little bit about how that might be shaping your decisions regarding manufacturing investment and what kind of measures you could use to reduce costs, for example?
So first of all, owning our manufacturing facility allows us control over time, control over quality, control over cost improvements of the TIL process. So that's an important lever that we have at our disposal. We are focused on several areas. One is optimizing our workforce, optimizing our capacity utilization to make sure that ICTC is a cost-effective manufacturing site. And longer term, we have initiatives ongoing for process improvements and streamlining the manufacturing process in multiple areas. And then automation that I mentioned earlier, that's an important long-term lever to further streamline this the till manufacturing process and further drive manufacturing costs. And the experience we have right now from the expanded access program for EAP is also important for testing all of these processes in the commercial setting and ensure that we minimize any surprises that may affect costs as well when we go into commercial manufacturing.
And so on the automation front, can you expand a little bit on how that might be employed?
So there are several areas. Some are very near term. It's around supply chain and the cell journey through various steps of logistics in the manufacturing process. The other area is around scheduling and supply chain logistics. And longer term, it's around automating various steps of the manufacturing process further, which in a way we're somewhat following in the footsteps of other cell therapy companies who have been on the market for a few years now, and we've been hearing they're taking such steps. So, and that's what we're planning to integrate potentially in the next stages of build out of our facilities, whether it's the shell space we have at ICTC right now, or a new facility that we may be putting in place. And so the goal would be to provide as much automation into those new facilities as possible to maximize their capacity per square foot and improve also efficiency of the manufacturing process.
Great. And we've heard about some supply chain constraints in the cell and gene therapy space in recent years. Can you tell us how you're managing supply chain in your facility and what you're seeing these days?
That's an important area. We've been taking a number of levers to make sure that the supply chain constraints do not affect our manufacturing. And that includes having redundant suppliers, having commercial supply agreements with some of the key vendors, stockpiling inventories when necessary, and other risk reduction approaches. And one example that has been coming up in the industry is fludarabine, cyclophosphamide supply chain shortages. So our team's done a great job keeping a close eye at that and making sure that those industry shortages do not affect our clinical trials or our patients who are receiving TIL. Great.
I do see an audience member question regarding the overlap between the trial sites and the ATCs that you have planned. Any comment on that front in terms of the overlap between those?
So we're planning for about 40 authorized treatment centers around the time of launch. And that's based on the concentration of patients that we've seen in the CAR-T space, where top 40 sites are accountable for about 80% of treated patients. And in melanoma, we expect patient concentration to be fairly similar. And some of the 40 authorized treatment centers, we expect to be the ones who are currently participating in our clinical trials. There's a slide in our corporate presentation that shows the locations of those sites, future treatment centers for commercial and current clinical trial sites. And there's a very significant overlap. The other ones we're selecting based on their being NCCN treatment centers, locations where there are major KOLs, as well as to ensure that there's good geographical coverage for the patients to provide convenient and easy access for them for TIL therapy at launch. And the training program for each of these sites is, of course, customized based on their prior experience with TIL therapy, whether they've been working with us on clinical trials already or whether they're relatively new to TIL.
Got it. Okay, great. I see we're just at the end of the session, so I'd like to thank you again, Igor, for your participation and for fielding our questions.
Cool. Thank you very much. Thanks for having us.